Lecture number 20. Haploids.
यार अभी लाइट जाना पंखा भी और ये इधर आओ जैनिमल उठाओ
Haploids. Introduction. A haploid is a cell or organism that has a single set of chromosomes that are not paired. The haploid gamete is normally produced during plant cell division. During fertilization, these cells normally merge with other similar haploid cells. A haploid cell only has half the number of chromosomes as are present in diploids. Applications of haploid plants. In vitro production of haploids can also solve some problems in genetic studies. The following points highlight the top applications of haploid plants. Development of pure homozygous line. In plant breeding, it is very much essential to get the pure homozygous line, which is generally obtained through selfing for 6 to 7 generations, but by the use of anther or pollen culture, it can be reduced to few months or a year. These genetically pure homozygous lines are used for breeding as well as genetic research purpose. This technique is also helpful for breeding of these plants which have more elongated juvenile phase. Selection of mutants. Resistance to diseases. Mutants with resistance to disease is of prime importance in crop improvement. Haploids provide a relatively easier system for the induction of mutations. Some examples of using anther culture technique in mutant successfully are tobacco mutants resistant to black shank disease and wheat lines resistant to scab. Transfer of desired alien genes. Chromosomal instability in haploids makes them potential tools for introduction of alien chromosomes on genes during wider crossing programs. Induction of mutagenesis. Haploid cell cultures are useful material for induction of mutations and to study the effect of mutation. This method can overcome the masking effect of presence of dominant gene. The screening method for detection of mutational effect is also easier in this technique. Induction of genetic variability. The pollen or microspore are easy explant for production of genetically variable types by introducing the different foreign genes through different transformation procedures. These transformed or transgenic haploids can be used further in breeding programs. Development of aneuploids. Haploids have been used in the production of aneuploids like monosomies in wheat, trisomies in potato. In tobacco, nullisomics were derived from haploids obtained from monosomies which could not produce nullisomics on selfing. Nullisomic is a genetic condition involving the lack of both the normal chromosomal pairs for a species 2 and -2. Thank you.